Okay, good evening, students. Today we will be starting the first class of this semester, and I will be teaching you prokaryotic genetics. So, the first topic of this subject is mutation. So, what is a mutation? Mutation is the sudden heritable change in genetic material. This is called mutation. So, mutation is sudden and mutation is heritable. Individuals showing changes after mutation are called mutant, and those who don't show any alteration are called wild type. And the factors or agents that are causing mutation are called mutagen. Today is the introductory class, and I will be discussing only the basic things about mutation and all. And in the next class, we'll be going into the more details on mutations and topics. So, what are the characteristics of mutation? So, when we study mutation, some things we have to remember is the mutation. Generally, the mutant alleles are recessive to their normal or wild type alleles. So suppose in human beings, we have two alleles, one from my father and another from our mother. So one allele is wild type or unaltered and another is mutated or changed genetically. So remember that generally the mutant alleles are recessive to their normal or wild type alleles. Next point is most mutations are harmful. Most mutations. They are causing cancers and all and many diseases, but some are beneficial. Spontaneous mutation occur. Actually, spontaneous mutation occur, but they occur in a very low rate. Some genes show higher rate of mutation than other genes. Such genes are called mutable genes. And highly mutable sites within a gene are known as hotspots. So hotspots are positions of a certain gene where mutation occur more frequently than other sites. So if we want to classify mutation or divide the types of mutation, we can divide them on the basis of many characters, like based on causes of mutation. Like it can be spontaneous, like it is occurring naturally without any cause, but the rate is very low in case of spontaneous mutation, because spontaneous mutation occurs independent of anything, but they occur in a very low rate. And another is induced mutation. What is induced mutation? This is a type of mutation that occurs due to some physical or chemical agent. Suppose some X-ray, some gamma ray or something else, and some chemical agents. So they may caused by some physical or chemical agents. So they are induced by something. So they are called induced mutation. Another classification based on the tissue of origin. Somatic mutation, if the mutation occurs in the somatic cell. Another is germinal mutation, if the mutation occurs in the germ cell or sex cells. And based on direction of mutation, it may be forward mutation or it may be reverse mutation. What is forward mutation? That is a general mutation where the mutation from the wild type to the mutant type, while the reverse mutation is a reverse from, that is a mutant to the wild type. So types of mutation, if we want to study that how many types of mutations there may be chromosomal mutation, like the change in the whole chromosome. It may be some part of the chromosome may be deleted, it may be duplicated, it may be inverted, or it may be translocated from one place of the same chromosome to the other place of that same chromosome, or it may be translocated from one chromosome to the another chromosome. So these are chromosomal mutations. And another is gene mutation. Gene mutation means these are change in the genetic sequence of a specific gene. So gene mutation and chromosome mutation, both are very, very important. Gene mutation are of different types. Number one, point mutation. What is a point mutation? It is it is a change in a single nucleotide of a gene, or maybe two, or maybe three, but point mutation in generally refers to single nucleotide changes in a gene. And the point mutation is of three types. One is silent mutation, missense mutation, nonsense mutation. Now, what are silent mutation, missense mutation, and nonsense mutation? Silent mutation is, suppose a nucleotide is changed due to mutation, but after mutation, the codon, after changing the codon sequence, it codes for the same amino acid or similar type of amino acid. These are called silent mutation. So the mutation is occurring, but there is no change in the amino acid sequence or the function of the gene product. So we cannot sense it. So it is silent. Another is missense. Suppose a codon codes for lysine, that is a basic amino acid. If a mutation occurs in such a way that the codon changes from lysine to aspartic acid. Now, aspartic acid is negatively charged acidic amino acid. So, due to this mutation or due to this point mutation, the codon changes, and the resultant amino acid due to this codon change is totally different character. Before it was basic lysine, and after mutation it leads to the formation of acidic amino acid aspartic. So the protein, if we see the effect of this mutation on the gene product or that protein, it will be devastating. So this is missense mutation. And the last type of point mutation is nonsense mutation. If due to the point mutation, if a codon changes into stop codon, three types of stop codon we know. Okay, so if a codon changes into a stop codon due to a point mutation, that is called the nonsense mutation. And the other type of gene mutation is frame shift. So we can know the type or the character of this mutation from the name only. That is frame shift. Frame shift means it is changing the reading frame by which its nucleotide sequence will be read during protein synthesis, right? So it will be changing its open reading frame. So this leads to frame shift mutation. How can this be formed? Like if mutation causes insertion of some nucleotides within open reading frame, that can be a frame shift mutation, or it may be due to deletion of some residues of some nucleotides. This may also lead to frame shift. So we can see the types of mutation. Chromosomal mutation, they are of mainly four types: deletion, duplication, inversion, translocation. And gene mutation. Gene mutation is of two types: point mutations. Point mutations are of three types: silent, missense, nonsense. And frame shift mutation, that is of two types: insertion and deletion. So we are studying prokaryotic genetics. So we should not, it's not like that we should not study about chromosomal mutations and all, because it is needed to clear our view about mutation, what mutation can do. In the next class, I will be getting into the deeper understanding of prokaryotic mutation. Okay, so chromosomal mutations, how many types of chromosomal mutations are there? Number one, numerical changes in the chromosome, like aneuploidy. That is the abnormal number of chromosome. What is that? When an individual either is missing a chromosome or has more than two chromosomes of a pair. Suppose we have one pair of chromosome. Okay, one from our father and another from our mother. But if someone have more than one, more than two, three or four, these are abnormalities. So these are numerical chromosomal mutations. Examples, very important example of numerical chromosomal mutation is Down syndrome or trisomy 21. That means the patients have extra 21st chromosome in their body. It leads to impairment of both cognitive ability and physical growth. It is a very serious condition, no treatments available. Number two, Turner syndrome, when a female is partly or completely missing an X chromosome. We generally have 46 chromosomes, while in Turner syndrome the female has only 45 chromosomes. So these are the abnormalities in the number of chromosome that leads to severe impairment of cognitive ability and physical growth. Number two is structural chromosomal mutation. The first one is the change in the number of the chromosomes, and number two is the structural chromosomal mutations. What can be the types of structural chromosomal mutations? We can see from here, this is the chromosome, the green one, and there is a specific part of that chromosome that is deleted, and it leads to a smaller sized chromosome. So this is a deletion. It can happen in our body. Number two, translocation. Suppose the first chromosome, that is the green one, has a certain part, that is the red, and it can be cut and pasted into some other chromosome. In this case, that is the blue one, and thus by increasing its size, while the parent chromosome from where the translocation is occurring is getting smaller. So this is translocation. Duplication. What is duplication? A certain part of the chromosome is getting duplicated or doubled. So what happens next? The chromosome size increases, right? So this is duplication. And for this inversion, certain amount of the chromosome, certain things of the chromosome is what happening is getting inverted, so and then pasted into that the same position. So this is called inversion. So these are structural chromosomal mutations. So these are of mainly four types. Deletion, where a certain specific portion of the chromosome gets deleted. Translocation, a certain portion of the chromosome gets transferred into some other chromosomes in some other parts of the same chromosome, that is translocation. Duplication, certain part of the chromosome gets doubled or gets duplicated, that is called duplication. And inversion, certain part of the chromosome gets inverted and pasted into the same place but in a inverted manner. So these are different types of chromosomal mutations. Next day, in the next class, I will be discussing more deeply on prokaryotic on mutation in the prokaryotic context. So this was the introductory class to mutation. Hope you enjoyed. Thanks.